our speaker today. It's very exciting to be hosting Ilya Nemenman from Emory University. Ilya is a theoretical biophysicist who worked on many topics in systems biology and neuroscience. He is broadly interested in information processing, learning, and adaptation in neural, molecular, and evolutionary systems. He has various honors and awards. I have a list here, but instead of reading those, I want to say something else. So Ilya's work has been very impactful in many ways through mentoring and through his research. So publications, his impact on the field and on his students, but also for the larger community of biophysicists. And I'll just highlight one thing, which is many students in biophysics today know that if there's one exciting conference where we all go to, it's the GBio section of the APS March meeting. But not many current PhD students know that that has not always been the case. And in fact, making that the case was essentially spearheaded by Ilya. And every time I'm at APS, which is the one meeting I go to every year and always find very exciting, is I'm kind of thinking about the various ways in which someone can have a far-reaching impact. So I'll just let Ilya talk about his science instead of me. And
thank you
for
having me here, and thank you everybody for coming. It's the first one talk that I'm given in any department in the last 2 years. I'm actually very excited to be here and just getting the energy of talking to everybody and just seeing people in the room. It's really that's why I'm in academia, more or less, right? It's for this moment. Anyway, so thank you everybody. I'm going to be talking about emergent dynamics and high throughput biological data. I will always, as always, I will run late. So let's start with the important part. The work was largely done by 2 people, Audrey Sederberg. Audrey was a postdoc and she is now a faculty member at the University of Minnesota in neuroscience, and Mia Morel was an undergraduate and is currently a graduate student in New York. So what I thought I would do is, before going to actual theories, data, and all that, I will try to give you some philosophy, like maybe even a bit too much. And so, but stay with me for a bit. So the first question first, not everybody here is biophysicist. So why it is that a physicist should study life? Why should physicists study life and brain in particular? Right? And so for me, this was the question that I answered for myself when I was about 13, when I ran into this quote by Immanuel Kant, that there's 2 things in the world that fill me with an unimaginable awe. That's the infinite depths of the starry skies above my head and the infinite depths of the moral thought inside my head, which in the modern language is, roughly speaking, how did the universe start and what is consciousness, roughly speaking, right? And so, you know, as a guy who knew some math, I decided I'm going to do cosmology first, and that didn't work out. And then, you know, there's 2 things, the second thing, right, that was in the world. And that's when I switched around middle of my graduate school and have never looked back, right? So, but the other question, I had this debate at some point with David Gross, you know, is biophysics, neuroscience, or more generally an interesting thing or not? And what I would argue is that, you know, studying string theory cannot be more exciting than studying brains that can study string theory, right? And so I think that those of you who know David, you can try to imagine sort of how that argument went. So what should the physicist hope to find when we try to study life? And here we go to yet another Nobel Prize winner, right? Not David, but in this case, even you know, maybe more important, von Schrödinger. And this is a quote from his essay on what is life: that living matter, while not eluding the laws of physics as established up to date, is likely to involve other laws of physics, hitherto unknown, which, however, once they have been revealed, will form just as an integral part of science as a form. And so that's my goal. This is the goal. This is what I want to see. I want to find new laws of physics inside biological systems. I'm not necessarily interested in curing a disease. I'm not interested in biology. I'm not a biologist, even though I'm in the biology department as much as in physics. but I'm a physicist, I want new laws. I want things that generalize from one system to another. And that's what you should have the eye for in all of the data, all of the models that I'm going to present you for. So what is different now? Why should we try to study this field now? Well, of course, right now, what we finally have is data, right? We have a lot of data. I'm going to show you examples of data, but something is still missing. And that's another one of my favorite quotes. This is a quote from a short story by Borges. In that empire, the College of Cartographers involved a map of the empire that was the same of the same scale as the empire and coincided with it point for point. And then things went wrong, and in the Western desert, tattered fragments of the map are still to be found, sheltering an occasional beast or beggar. And so I think that's where we're going with modern biology, unfortunately, is that we're getting more and more and more data. We're building these huge atlases of different systems, in biology. This is gene expression atlases in the brain, or different gene expressions in the different cell types in our body, or trees of bacteria as they relate to each other. And we're being drowned by data, but really don't understand what's going on. And this was probably put the best by Sid Brenner, one of the founders of molecular biology, one of the people who put us on this path to generate more and more and more and more data and put it in bigger and bigger databases, that biological research is in crisis. We're drowning in a sea of data and searching for some theoretical framework with which to understand it, right? So theory is missing. And that's what I will try to get us to, sort of some maybe simple theoretical ideas, how to interpret biological data and what these theoretical ideas are going to give us. So why is theory missing? In fact, on microscopic scales in biology, are just physics, right? There's some molecules, they interact with each other, with the potentials, the laws that we already know and love, right? The same quantum mechanics or classical mechanics, electrodynamics and all of that, right? There's no life force as far as we know, so everything down there below has to boil down to physics, right? And in fact, it works. So this is the equations from the Nobel Prize winning Hodgkin and Huxley papers describing propagation of an action potential in an axon. This is just Ohm's laws, right? With some voltage dependent resistances, right? And it works perfectly well. So what is different? Why couldn't we take this sort of simple laws and describe the entire brain that we have, right? And the problem is that these laws do not work for systems which have millions of heterogeneous and poorly known components. We can describe the propagation of an action potential for a single axon, but when you have a brain that sort of looks like this, where there is this type of equations for every tiny compartment, of different colors, this is actually the actual wiring diagram of certain slice of the brain of mouse, if I remember correctly. Don't quote me on this, right? Then you will have to have the same equations for every tiny compartment of the system. And then, if you're going to have an Avogadro number of compartments, then this is just not going to work. You will need the sort of planetary size computer to model this type of systems, and you will still get things wrong because you don't know all of the parameters to a high accuracy, right? So this is a problem. There is even a bigger problem that in a lot of systems, the degrees of freedom for these systems are not well known. So for example, in ecological systems, which can hope to describe with very simple equation, this would be like the basic equations for interactions between species. We don't know what species are. This is actually what Misha works on or used to work on. What are the variables that we care about to measure? We do not know that. In physical systems, we know that we need to measure mass, and then you know that F is equal ma, and you know how to calculate acceleration from, say, force. In these biological systems, I do not know what is the right quantities to measure. I can measure everything, and yet I don't know what to measure. And that's where I think the biggest problem is. So why should there be new laws? And again, I'm not going to invent a wheel here. I keep on quoting important people. More is different, right? This is Phil Anderson. When you put a lot of things together, some new statistical laws are going to start to emerge. And why is that? It's because we have various forms of generalized central limit theorem. Right? You put a lot of things together, you normally get a Gaussian. Well, except that if things are correlated, you don't get a Gaussian, you get something else, generalized central limit laws. And so you get central limit plus locality plus symmetry that constrain what it is that you're going to get, and you get universal effective descriptions, and there are not that many of them, right? And once you get a universal effective description, at that point you should start asking what is the simplest possible way to describe that probability distribution, that joint activity that you got. You know, mean and variance would be, if we're talking about a Gaussian, and maybe for different systems it's not going to be a mean and variance, it's going to be something else. And those are the things that Kadanov and Goldenfeld were talking about, that you do not model. You don't want to model the bulldozers with quarks. You want to have the correct degrees of freedom that are properly tuned to the scale at which you want to model. And I'm giving all of this stuff, and we're going to get to some of the examples where I think there is maybe some hint that we are going to see these new degrees of freedom emerging in biological systems. So biology certainly has more, right? I said that more is different. So this is the typical data set that we deal with in biology. This is the activity of a bunch of neurons in a, I think it was the hippocampus of rat, I may be wrong. And the idea here is that time goes on the horizontal axis, neuron number goes on the vertical axis. You have a spike, an action potential at a certain point, you get a black dot. If you don't have anything, you get the white dot. And so we need to build a theory of something that looks like this. Right? This is the more that hopefully we can use from Anderson's code to create something different out of this more. But the interesting part here is that essentially any modern high throughput biological experiment is going to have data that looks very similar to this. So if I were to, for example, go and sequence, or if I were to look at a single neuron, and I'm going to do a typical experiment, not many neurons, but a single neuron, a typical experiment that people do in the lab, which is to show the same stimulus to the neuron again and again and again and again and see how it responds. Sometimes it's going to respond to the one stimulus and sometimes it won't respond to the same stimulus. You will get the same raster diagram where, you know, over here it's going to be time and this is going to be a repetition number for that one particular neuron, right? Or you can look at, let's say, structure of proteins. And there is a sequence of a protein and there are related proteins that perform the same function. You find them in the protein data bank. And you're going to see at a certain point, there is a consensus amino acid, and some protein is going to get a different one at that point, and some protein is going to get a different one at a different point. And so what you're going to have is, this is a position in a sequence within this protein, and on the vertical axis is going to be a species number. And we're going to have a dot if there is a mutation for that position, for that species, or we're going to have no dot if there is no mutation for that position, right? And again, all of these rows are going to be just like for neurons. All of these rows are not independent because neurons talk to each other. They're responding to the same stimulus. They're talking to each other directly. In the proteins, it's going to be the same story. The dots are not independent because the proteins perform function, and there is allostery and there is joint evolution across multiple sites and so on and so forth. You will have the dots and the black dots and the white spaces distributed through some weird correlation structure. You can look at gene expressions and the same story. This would be a gene ID, and in one cell this gene is active, or the chromatin is accessible for expression of this gene, and some other cell it's not active, chromatin is closed, and you get the same zeros and one pattern for describing the dynamics, for the state of the gene expression of the cell, right? So as far as I am concerned, when I look at the data, biological data, this is how I see it. I see it as a bunch of zeros and ones in this big table, usually two-dimensional, sometimes three-dimensional if you have multiple different conditions. And what I'm interested in understanding if what can we say about the statistical properties of the joint probability distribution of these big tables, right? of data in these big tables. Are there any interesting central limit-like behaviors that we can calculate or can measure? Okay. So let's look at some of these examples. I'm going to start with one. I'm going to give you a few more later. I'm showing 4 different completely independent experiments. So the first experiment, this is sequences of antibodies in B cells in fish. So now after COVID, everybody knows what antibody is, so I don't have to explain that, right? Antibodies are slightly different. And so what we're plotting here is the most common antibody comes first, the second most common antibody comes second, the third one comes third, and so on and so forth. And then we plot the frequency of these antibodies in the entire blood of this poor animal versus the rank, right? How common it is. So it's rank frequency versus rank plot. And you see that this is 1 over rank, frequency equal to 1 over rank, what's called the Zipf's law in languages. And you see that the system is actually following the Zipf law pretty well. That's for one animal. There is 10 other animals here, 1 over F law. They all are on top of each other. So there is interesting Zipf's distribution of rank versus frequency in antibodies. This is a totally different experiment. This is salamander retina. And we're looking at activity of a bunch of neurons. And the neurons are sometimes going to be inactive zeros, or sometimes they're going to be firing ones. And I'm going to find the most common combination of these neurons, which in this case is going to be everybody's silent, nobody's firing, 0, 0, 0, 0, 0. And I'm going to count how many times does this happen, right? And that's the first entry in this plot. And I'm going to find the second most common combination, the third most common combination, and plot their frequencies on this plot. And you see this dotted line is 1 over rank. And if you look at groups of 20 neurons versus 30 neurons versus 40 neurons, by the time you get to 40, you're very close to 1 over rank. And so you can extrapolate that maybe if I could record, which this is an old paper, now we can record from more than 40 neurons at a time, we'll probably be at the Zipf law and we will be fine. This is a single neuron in a fly. This is a neuron which is responsible for detecting horizontal motion, self-motion of the animal. And what we're going to do, we're going to track this neuron over time. We're going to look at this piece of time series of its activity, zeros and ones. And we're going to look at the most common one, which again turns out to be all zeros, right? Neuron is not firing half the time. Then we're going to look at the second most common, the third most common. So it's now not multiple neurons over time, but a single neuron, not multiple neurons at the same time, but a single neuron over time. And you get the Zipf law, the black line over here. And what you notice is that if you look at short words, like the durations of activity of about 20 milliseconds, and this is a green line, it's not Zipf, but by the time you get to longer words, you start having a Zipf law. This is sequencing of certain proteins in HIV in the body of a patient. This is GAG and NEF proteins. And again, the same story as above. We're looking at the most common protein, variant of this protein, the next most common, the next most common. This is 1 over F Zipf law. Okay. So everywhere, these data are as diverse as they could be, and yet we're generating a Zipf law. And when you see this, there is a hope that maybe it's kind of a law, right? There's something going on that we haven't quite figured out, which is not specific to neuroscience, which is not specific to immunology, which is not specific to protein evolution, right? So the same thing, I showed you just 4 plots, but you can see exactly the same statistics for various data in gene expression, in chromosomal accessibility, in cellular mechanics and motility, in microbial compositions. It's the Zipf law pops up every single time. And so the question is why? What do this data, that this 1 over F, 1 over rank, right? Frequency is 1 over rank. What does it tell us about the underlying system? Okay, so that's what we're going to try to explore in the next few minutes. So this is the emergent law that we will try to explore, and why it is that this is sort of would be Anderson's different, right? More is different. We put this very large recordings from 20 milliseconds. There was no interesting law, and at 40 milliseconds, you're now getting, interesting laws showing up, right? So more should be equal to different. So why has it been difficult to find what this different is? And the problem is that traditional theoretical physics tools do not work well on systems where there is no symmetry, no locality, no homogeneity. Intuition that we have about such systems fails. most people argue that, would argue that when we get this beautiful 1 over f law, it tells us something about the system. Maybe the system is poised to criticality to perform some interesting computation, etc., etc. I've talked to many of you here about this over the last couple of hours. But in reality, what I would argue that you will almost always get from this from totally unrelated reasons, right? Our intuition fails when we deal with systems like that, because we're not trained to look at systems that are not local. And we don't know what to expect from experiments. We don't know how to be surprised or when to be surprised. And that's why we actually maybe measure things that we should not be measuring and miss things that we should be measuring, right? And so how do we move forward? How do we try to distill this different from this very large data sets? And so my thoughts about this are, what I will try to show you today is, at least in the few corners of this high throughput biological data analysis, biological data sets are fitted exquisitely well by models of random network, purely random networks with a few biological constraints. And those constraints are important, and that's what makes this network behave slightly differently. But nonetheless, random networks. And it's not that we are talking about symmetries of a specific network, but we're talking about symmetries and structures of ensembles of possible interactions. And so I'll try to explain this. What I will also try to show is that the simple models are actually universal across different biological domains. Some of these models are equivalent to existence of relatively low dimensional, hidden or latent, as they're called in the field, degrees of freedom, and these degrees of freedom are possibly emergent, right? They are a collective effect of an activity of all of these units together, and they describe the interesting part of the dynamics almost completely, right? There is nothing interesting and remaining in the dynamics besides this emergent collective degrees of freedom. And unfortunately, to date, we still don't have good prescriptions of how to extract these latent dynamical features from data. I'll show you a few examples where we maybe were successful because we guessed right, but that's where most of research nowadays in my group is in trying to identify this emergent features in the data. That was the introduction. Yes.
I have a very naive question. Yeah. So can you give me a intuition, what is the microscopic law that governs all the small particles? Are you thinking about quantum mechanics or more like a classical physics? In the microscopic law.
I mean, if I go further down, there's probably some string theory somewhere, right? And it probably is not relevant completely, right? And I think that for what I'm going to show, it really absolutely doesn't matter whether quantum mechanics is important, whether it's whether it's true mechanics, whether it's ENM, it really doesn't, right? But somewhere down below, everything is governed by strings, right? It's just not the right level of description. It's sort of, again, Goldenfeld and Kadanoff, you do not want to model bulldozers with quarks, so you don't want to put quantum mechanics on a whole zebrafish, right? It's just, you're never going to be able to calculate it.
I thought somehow recently people are claiming, even in your brain, somehow the nuclear spins or something.
Yes.
Different isotope of nuclear spins actually matters, and you end up having quantum mechanics dominating everything.
Absolutely.
But I don't know. I was just curious.
Yes, absolutely. People are claiming that.
A brain is like a quantum computer, but somehow you just don't know.
Absolutely. Yes, people are claiming that. I remain to be convinced.
I see. I'm just curious. Thanks.
Okay, so what I want, before I actually go further to actually show experimental data, I want to try to show to you just one example of this random matrix universality. We'll talk quite a bit about random matrices and how when you put them together, you get this interesting universal results. And so before going that, I will just talk about something that is extremely well known and something that almost anybody in this room who works with data has done, right? So suppose you are recording data like this. This is just this big matrix that I showed you, where you have 100 variables and you are recording them 1,000 times, right? And these variables could be binary, they could be continuous. It really doesn't matter for what I'm going to say. So I designed this data. There's no correlations between the columns. all of the variables are completely uncorrelated from each other. So if I were to calculate correlation coefficients, they should be 0, except that they will not be zeros because there is always noise in the data and you will get this spurious correlation effect. So the question is, if I calculated the covariance matrix between these 100 variables with themselves, what would be the structure of the covariance matrix? Okay. And so let's suppose I'm going to start first with 10 variables measured 30 times. I'm going to calculate the covariance matrix. I'm going to get eigenvalues of this matrix, and I'm going to plot them in a histogram, and you will get these are two different implementations of the same data. This and this, they don't look anything similar to each other. It's just, 10 eigenvalues and some other 10 eigenvalues. And I go to a larger matrix, so 100 variables measured 300 times. And I'm going to calculate 100 by 100 covariance matrix, take its spectrum and plot the spectrum, and you start seeing some similarity. By the time I go to two different realizations of 1,000 variables over 3,000 times, the two spectra look essentially equivalent. Right? This is the celebrated Marchenko-Pasteur spectrum for what's called B-sharp matrices. And so I think that from my perspective as a theoretical physicist, this is one of the biggest disservices we're doing to us, to education of next generation of physicists, that we are not teaching random matrices, like Wigner's idea of the spectrum of heavy nuclei calculated through random matrices and so on and so forth. We're not doing that, and that's a big and largely forgotten part of the 20th century physics, which nowadays is starting to reemerge again in statistical physics, in condensed matter physics, and in biophysics, as I will point out. So basically, law of large numbers, when applied to matrices, can produce very interesting and really non-Gaussian structures, right? It's just that this matrix had this that we're working with was a covariance matrix. It's not just random entries, it's a covariance matrix. How many of you actually have done this in your labs, right? Done PCAs and like all biophysicists, presumably, right? Okay, so let's go back to the zip flow, and then we'll relate it back to the random matrices. So the first thing that I want to point out is that this zip flow is actually a very special notion of a physical criticality. This was calculated by Thierry Morin and Bill Bial a decade ago. So if I have a sequence of spins, zeros and ones, this would be my vector sigma. And what happens if this probability distribution of absorbing a certain word goes as 1 over the rank of this word to some power alpha? What does it mean, right? And so what they've been able to show is that for this system, then the energy, which is the log of this probability, is just defined as the log of this probability. And then the energy density logarithm of the energy density, which is the microcanonical entropy, become proportional to each other. It's just a 3-line calculation. But if this is the case, then the quantity which we call the heat capacity, which is the inverse of the second derivative of entropy with respect to the energy, becomes infinite. And infinite heat capacity means phase transition criticality and so on. So whenever we see a zip flow, this is a very special notion of criticality, right? The system is critical by physics definition, but it's a very special criticality. Yes?
Are you using rank because you don't have a metric or a distance between two different things, and that's just because there's no natural?
There's, this, as I said, this data are not local. We're recording from neurons. They can be local in space, but it's very easy for a neuron to run an axon to the other side of the brain and not to be connected to the nearby neuron. And so physical locality doesn't mean that they are active in some local sense. It's going to become relevant in maybe 20 minutes, 15 minutes. Hopefully earlier than that. I'm looking at the data, at the clock. Okay. So it turns out that if you write a very simple model, and this is the only equations I'm going to show in this talk, so let's just walk through them. So we're going to have my spins. They're all conditionally independent given some external unobserved latent field, right? It's H. And so probability of sigma given H, they're independent, so it's a product of over them. And but after that, I'm going to allow my external field, the latent field, to fluctuate. And so I'm not going to observe the spins at a particular value of the field, but I'm going to average over the field, so marginalize over the field. So the actual distribution of H's is just an integral of this object with respect to the unknown to me probability distribution of H. So it turns out that in this system, whatever that probability distribution of H is, as long as the value of the spin, the magnetization, is actually susceptible to the H. So when you change the H over the range in this probability distribution, and the magnetization actually changes by order of 1, as long as this condition is satisfied, you get the zip flaw. That's it. You cannot avoid it. Right? So this comes from a very simple application of large deviation theory from Gardner-Ellis lemma, or you can revert it back to what's called a symplectic equipartition that Shannon calculated in the 40s when he was developing his information theory, communication theory. Yes.
Curious again, I'm not familiar. So does this actually depend on dimensionality of your system?
Yes and no. It depends, of course. So what we
Usually I compare the alpha, the power law, with the dimensionality. At least that's what typically I do in most of my things.
So here I would get a zip flaw exactly with alpha equal to 1. If this method, if this is the reason if this is what's going on, then alpha has to be equal to 1. So it's 1 over rank, not 1 over rank to some power. Right? And so the only thing that are required is that each individual unit is susceptible to the external stimulus, to this H, and what's also required that the number of units is large. And remember what I showed to you is when I have 20 units versus when I have 40 units, we start looking more and more and more like the true zip flaw. So the conclusion here is actually kind of simple, that the existence of zip flaw, and especially if you're approaching zip flaw as the system size grows, means that the system has latent fields. And that's it. It doesn't tell us anything about the underlying interaction between these fields and the system. Any kind of latent fields, they don't have to come in with the simple linear interactions that I showed you on the previous slide, will result in the zip flaw. And again, intuition has failed us, right? We thought that we would, that when you introduce some kind of extra coupling between independent units, you will produce rank ordered plots, which may depend on what the statistics of the field is, but it doesn't. You get the zip flaw all the time. We cannot say much about the underlying system besides that the system is adaptive in the sense that its units are susceptible to the H's, right? The H changes and the activity of the units actually changes as well. Right? And this is here, that's the same fly neuron that I showed you. The black data is the same data which I already showed you, and the blue is the model that with no adjustable parameters, nothing, this is what it would reproduce if you just assume that there is one simple latent field in the system, and this latent field drives the activity of this neuron and changes over time. Yes.
All the examples, the variables that you're considering are discrete.
Yes.
So usually in physics, we measure sometimes momentum, which classically is a continuum method, right? So you only, this zip law applies to, is that a limitation of your?
Yes and no, right? Of course, if you want to plot rank or the frequency of occurrence of a certain word, a certain state, the state has to be discrete. Otherwise, you cannot really calculate the frequency of occurrence of a certain state in data, right? It has to be discrete. So you can discretize your continuous data, and as soon as you discretize continuous data, you'll get the same thing back. But yes, if the data is truly really continuous, I don't know what's going to happen. So this is so far was all of the data that I showed you is what I would call sort of one-point statistics, one point in time, right? We just looked at the statistics of activity of neurons or expressions or sequences and so on. But physics also has dynamics. And so what would happen if you get a model that looks sort of like this and allows the fields to vary with time? So let's look at the data. And here we're going to connect again to yet another beautiful part of physics, which is renormalization groups. This is work was done by Nemenman and Bill Bialik about 2 years ago. So the experiment here looks like this. This is a mouse. Mouse is running on a ball. It's immobilized, and its brain is open. There is a glass there, and you can see what the neurons are doing, and you can record the neural activity. And this is what it looks like. There is about 1,000 neurons in that field. These neurons are active. We can record zeros or ones when they spike. And the mouse runs on a track, right? It runs on this ball, but there is virtual reality around it. And so the mouse believes that it's in a maze. It's making, goes forward, and then it's being immediately transported back, and it keeps on running. The mouse doesn't have a certain part of the brain there, so it really doesn't even feel that it's tired and all that. It just keeps on running forever in this maze, and we can record a lot of it. So we can record thousands and thousands of runs through this maze from thousands and thousands of neurons nowadays. And so what this group decided to do is they wanted to understand, what does this data have some kind of collective description? Right? How do we get collective descriptions? Well, we have only one way of doing this that we know of, but this is applying renormalization group, right? We're going to coarse grain the system and see how it starts looking like as a function of the size of the coarse graining scale. So what they decided to do basically, but when we are doing, let's say, Kadanoff's renormalization group, we are combining two nearby spins. There's no nearby here, right? There's no spatial locality. So what do we do instead? We say that things which are active over time in a similar pattern are in some sense activity local, right? They're close to each other. And so we're going to combine similar looking units with each other. And so I'm going to look at neurons, 1 and 2, their activity over a long time is similar. I'm going to combine them into some superneuron or hyperneuron, hyperspin, and so on and so forth until I get down to clusters. A single cluster is going to have many neurons in it. And what do we expect at this point? As the clusters get larger and larger, we can evaluate correlations between these clusters. And we would expect that correlations between these big clusters should start falling at a certain scale, maybe, right? What we would expect is that these clusters, the temporal activity of these large clusters, because many units are contributing to them, right? There's just a lot more noise, they will decorrelate over time quicker. If my clusters are sums of neurons, remember that variances of independent variables add, right? And so what it means that if my neurons were really independent, then the variance of the clusters activity would go linearly with the cluster size. But if my neurons were all equal to each other, perfectly correlated, then the standard deviation would go linearly with the system size. So the variance would go as a square, right? So we expect the variance to scale as somewhere between 1 and 2 maybe in this system, right? And if we're adding a lot of things together, we should expect a Gaussian random variable central limit to show up. So the general activity of the cluster, which is a sum of multiple neurons, should become Gaussian. So what they observed is system is neither uncorrelated nor fully correlated, sits somewhere in the middle. Variance of the cluster activity, eigenvalues of covariance matrices has this beautiful scaling law, and the correlation, the correlations between these clusters become slower and slower with time. But there is again a beautiful scaling that the correlation length is dependent and grows with the cluster size. And if the cluster presumably goes to infinity, the correlation length becomes infinite, right? So the system is really critical, right, in that sense. So this is what they observed. This is what we did. We did the same model that I just showed to you before. A bunch of neurons, a bunch of scaling fields, latent fields. In this case, only about between 5 and 10 scaling fields and about 1,000 neurons. And we're going to impose biological constraints on the system that is that this system has place fields and place neurons and non-place neurons. It's not important for this audience, but for neuroscientists, this would be important. there is about 50% connectivity in this network. Again, not important for this audience, but for neuroscience would be important. There is no learning, no adaptation, nothing, but just randomly picking up kind of interactions between this and this units. And we're going to allow this latent fields to undergo Ornstein invariant dynamics, based random walk dynamics with a correlation time, with a single correlation time for all of them, which is much longer than the activity of an individual unit. This is what you get. I'm showing the exponents that we generate in models and the exponents that they measured in theory on the previous plot, and they agree with each other to significant digits, except for the time exponent, which we know why it doesn't. So I'm not going to go into that picture, but you get this beautiful scalings just from the model of completely non-interacting or conditionally independent neurons. Okay. We're also going to explain a bunch of other things, like, for example, how does the overall probability distribution of activities of these clusters look like as I make clusters larger and larger? This is from experiment. This is log of the probability versus the activity. This would be a Gaussian, right, in the middle. So if the central limit was holding, and it's clearly not Gaussian. This is from our models, from other simulations. This would be the Gaussian. And you notice that even the shape of the distribution is correctly modeled here with the same shoulder on the right-hand side as here. The scales here are different, so just pay attention to that. Overall, these two plots are essentially the same. Right? So it's not even that the scalings are there, but the entire probability distribution of clusters activity in these experimental data sets are explained by this completely independent model, right? So what it means is that there is nothing interesting in this data set to some extent besides the activity of the underlying latent variables, and then all neurons are just driven by those latent variables. They just go along for the ride. That's it. There is nothing interesting going on with them. It's just the latent activity that drives the dynamics. We haven't yet been able to prove this analytically. We've only been able to show this numerically, but stay with us for a couple more years. Analytics is always hard. So the recap here is that many of the scaling laws can be explained by these random models with dynamical latent features. Our intuition, and there are other things that can be explained. I already talked with a few of you today that the things like hyperbolicity in neural data or the avalanche scaling in neural data, which are beautiful fields in their own rights, also follow from these very simple models with numerical precision, right? Quantitative precision. So our intuition clearly is failing us. And these are emergent universal laws, right? This latent feature that I'm talking about, not the neurons, are the system, right? The neurons just go along for the ride. They are boring if you condition them on the activity of this latent variables. So the next question that we can ask, and it's the last thing that I'm going to ask, is where do these latent features come from, or could come from? Of course, I don't know the answer for every of these problems. And also, can these models with latent dynamics perform interesting computations? So far, I just showed to you that they perform, that the statistics matches, right? But can they actually do something? So let's look at another experiment. This is from Anne Chorchland's lab. It's a mouse. Mouse is hearing clicks. It's a Poisson sequence of clicks. Is a 10 Hertz Poisson or 20 Hertz Poisson, and it needs to make a decision whether it's hearing 10 or 20 and needs to go left or right. And what we're going to be doing, we're going to be measuring the activity of the brain, a certain part of the brain in the mouse, before it makes a decision to go left or right, and we'll try to predict whether which way the mouse is going to go, which way the decision is going to go from the activity of the neurons. So in this field, there is a standard model. The reason why this experiment was done is that there is a standard model that there is excitatory cells and inhibitory cells. Excitatory cells, like what controls, makes a decision. Inhibitory cells just keep everybody firing at bay, right? So you do not really fire either too high or too low. And so the main surprise for experimentalists coming from this model was that, from this experiment, is that this model doesn't work, because they could separately tag both excitatory neurons and inhibitory neurons. and they were able to predict equally well whether the animal is going to go left or right based on the activity of either excitatory neurons or the inhibitory neurons. And so it was a long process, and they built this beautiful multi-parameters, model of how decision-making happens in these neurons. And let's see if we can do something like this with a much less input into the system. So is there an alternative to this very structured models where some excitatory cells talk to some other cells? This would be the cells that are active when the animal eventually chooses to go left. This would be the cells that are active when an animal eventually chooses to go right, and they sort of suppress each other, so the winner takes all. Right? So what we are going to do, we're going to design a random network, literally a random network of interacting neurons with biological constraints. There is 80% of neurons are excitatory, 20% of neurons are inhibitory. And where we are going to show to this network two different stimuli. We're going to run the dynamics of this network, and we're going to try to predict whether this stimuli came from the stimulus 1 or stimulus 2 based on the activity of this network. And look at this classifier, which based on the activity of this network, makes a prediction. What is the problem? How does this classifier do compared to the experimentalist ability to predict where the animal is going to go in real experiments? Okay. This is how well we do. Again, it's a random network, but this is a random network with about a couple hundred neurons. So there's going to be some statistical irregularities. So some neurons are going to be slightly more active when the stimulus comes from the left versus when the stimulus comes from the right. And indeed, in this particular system, the sort of selectivity of each individual neuron, how much more likely you are to make the right decision if you look at an individual neuron, on average, is about 5% above chance, right? Which is true, in the real experiments that they measured is about 10% above chance, because they only measured from the neurons which are, there's a bias, right? They measured from neurons which are better than average, okay? But then if I look at my data and I try to build a classifier based on the activity of these neurons, did it go left or did it go right? And I do it many different times just to get enough statistics, we get that this network overall knows which way the signal comes from with an accuracy of about 80%, which is exactly the accuracy that experimentalists have measured. And I'm skipping a lot of other slides, but essentially every statistic that they measured in their experiments was explained by this model, including things like the probability distribution of weights in this classifiers, or probability distribution of time at which this classifiers becomes certain that it's making the right prediction in essentially every statistic that they measured in this paper. So why is this happening? So again, but now we're going back to random matrices that I pointed out. This is a random network. Random network, we can linearize it around its steady state. We can look at the spectrum of activity, and there are results going back to Wigner about where the spectra of the dynamics are going to lie. In this network, the spectra are going to lie in a big circle, right? So you will have oscillations if the things are away from the real axis, and you will have some modes that decay, negative eigenvalues, and some modes that grow, positive eigenvalues. And then I can look which, when my classifier makes a decision, what does it pay attention to? And what it pays attention to are these eigenmodes. And this is over here is the ratio of this to this, right? So it essentially pays most of the attention to random modes, which just randomly happen to grow with time, right? So there is some kind of excess of connectivity in this network. This neuron is slightly more active than it should be when it gets input from the left, and that neuron is slightly more active when it gets input from the right. It's actually important. It's not neurons, but it's a whole subnetwork of neurons, a whole mode of neurons, right? And that mode just keeps self-activating and growing up. And if that mode is active, the animal is going to go left. If the other mode is active, the animal is going to go right. And this is a result for any random network of this type, right? They will always have this distributed large mode that will predict where the signal is coming from. You do not need to tune it. And so, so what the important part here is that this emergent selectivity comes from existence of this collective mode, right? Which is a latent variable, right? This is a group of neurons that are going together, which would be the neurons that are talking to my latent field in the previous picture. For each type of stimulus, a different mode is going to be growing. For each type of stimulus, you will have a slightly different latent variable and a collective degree of freedom activating, but you will always have that in the system. And so what we've got here essentially is that this random network with constraint gave to us as a byproduct. First of all, it can do a computation. And it turns out that the way that it does a computation is by creating collective modes and then coupling these collective modes to the external stimuli, which are coming in to this network, so that an external stimulus activates the whole mode, right? So maybe this is where this collective latent variables that I talked about in the ZIF part of the talk are coming from, right? They don't have to be stimuli, they can be the emergent latent features. And so this is basically the parting thought at the end of the talk at this point. That first of all, what I hope I showed you is that we do have some new laws that are coming up, right? The ZIF law is everywhere in biology, right? So it's at least that, right? We have some new stuff that we haven't seen in physics before, and we now see in biology everywhere. And why? Because presumably in physics, we have systems which are closed, and in biology, they are open to all of the internal, external stimuli, and they want to respond to external stimuli because they are sensory systems, they live in the real world, right? So if you want to do this, you are required to obey the ZIF law. That's a statement, right? So current experiments that we've done cannot actually distinguish whether the network is designed or it's a random network, right? That's the last two slides that I showed you. What it means that we need to be looking for deviations from this ZIF laws and other types of statistics. What we need to be looking is at deviations in the predicted, computationally predicted modes of latent activity from experiments to see whether the network inside the brain is actually designed or it just happened to be what it happened to be, right? Is brain or other biological networks really random? Of course not, right? I mean, I would hope that my brain is not random, but it's interesting to see how far I can push this idea that most of the stuff that we are measuring can be explained as a null model, as just a random network with a few biological constraints. Okay? And on a deeper level, partly the brain has to be random, right? Because the number of neurons in our brain is 100 billion ballpark, and the number of genes is 20,000. There is no way that you can encode the 100 billion by 100 billion interaction matrix with just 20,000 genes. The best that you can do is to produce a bunch of general laws of who should talk to whom. And beyond that, it could be more than one law, it could be 1,000 laws, but nonetheless, rather small number, right? And beyond that, the connectivity has to be within those laws, within those biological constraints. It has to be random. Right? And so the other point is that what we've seen is that this latent dynamics is not only a description of biological data, but it's essentially a good compressed description, good in the sense that conditioned on these latent variables, neurons are boring, right? There is nothing left there. The variables are the biological computation. And so what this gives me a hope for is that maybe somehow we are on the way towards trying to do neuroscience without doing neurons, right? Trying to do neuroscience by identifying this new phenomenological emergent degrees of freedom and writing physics for those degrees of freedoms, how they interact with each other rather than how a billion neurons interact with other billion neurons. And similarly, can we do something about, can we write a model of evolution without talking about genes or species, but identifying collective modes in the genome space or in an ecological state of the system? How to do this, I don't know. Maybe in another decade, if I come here again, I can tell you something more. And with that, thank you very much.
I'm very curious. So the latent features, is one way to think about them, can they arise from just anatomy? So how would knowledge of the anatomy, so in neuroscience, maybe it's the connectome, in gene expression, maybe it's like the genome folding state. Like, to what extent would you be able to, I guess, uncover what these latent spaces really are, latent fields really are to knowledge of anatomy?
I don't know. I'm not trying to evade, I just don't know. It's clear that which specific latent variables you're going to get and how they're going to interact with each other is going to depend on the structures of these interaction matrices. And the anatomy is what's going to constrain these interaction matrices, right? So there is some mapping. I just don't know what it is. Like, you know, other people are doing this very similar stuff in the context of ecology. Like our common friend Pankaj Mehta, he has designed basically this type random models of bacterial ecologies, which seem to reproduce the bacterial species data in experiments with the same accuracy as our random models of networks, of neural networks do neural data, right? But the two data sets, the two models are extremely different from each other because the constraints that we're putting in on interactions are very different. Right? There he was talking about metabolites versus species. Here I'm talking about excitatory versus inhibitory neurons, right? And so I have no intuition at this point how, without doing the calculation or simulation, what the result is going to be, but clearly there is a dependence.
So fantastic talk, thank you. So there's obviously lots of different ways in which brains can go wrong, and you can describe that at all kinds of different levels. So I'd be interested to hear your opinions about the level of description you're talking about here. Do you think that will be useful for giving us insight into different ways in which brains can go wrong?
First of all, I am not a biomedical researcher, so I don't know, right? I think that some of these latent features are actually computational features, right? So for example, there is a set of neurons which light up when the animal goes left, right? And so you can probably see things where disappearance of a certain mode, a certain latent mode would signal an inability to do some kind of information processing, some kind of computation. How exactly, I don't know. There is actually a beautiful paper from here, from Hisyangen, right, from a couple of years ago on what happens to effectively, in this language, what happens to the latent variables in the ocular system that he works on if you put a patch on an eye, right? And you can see the difference. You can see that the dynamics changes. And I think it actually changes due to the same mechanism. We can almost predict it. Right? So there will be something, I just don't know. I don't have the intuition.
Mikhail, you tell us who.
Yeah, I'm wondering if what you've done is kind of like a cautionary tale to people interpreting experimental results. Like, I bet, isn't it possible you could find a lot of papers on a range of biophysics topics where people marveled at a certain power law or something, and you've actually shown that it's completely meaningless.
I would try to be a bit more optimistic here, right? I do think that it's not meaningless, right? So, for example, you will not get a zip flow if your system is not adaptive, right? If the system is not responsive to the outside world. So at the very least, what we've shown is that in all of these experiments, maybe it's not a surprise, but in all of these experiments, if you believe our theory, right, that the system is actually quite faithful to the statistics of the external probability distribution, so it actually responds to them. That's already an interesting statement, right? It would not be necessarily always the case. But even more beyond that, these latent variables could be of two different kinds, right? So one kind would be just an external stimulus, which is driving all of the neurons. And then, yes, you're maybe close to right in the sense that with all of the caveats about adaptation, et cetera, you're close to right in that it's maybe not the most interesting thing. But if those variables turns out to be not coupled to the external stimuli, but they're internal latent features, then at that point, like in an all-to-all Ising ferromagnet, you get an emergent magnetic field, and the system with a magnetic field coupled to the spins is mathematically equivalent to all coupled ferromagnet, right?
Kind of like self-organized type phenomena
so
where
if
just
I-
with
if
time
I-
they emerge. Yeah.
Right. If I can see that in these systems, the selecting variables are this emergent degrees of freedom rather than external field applied to this, then we actually got something. And that's what my hope is.
So I follow up on that. So if I think of a magnet, for example, then from the point of view of an individual spin, if you had these very long-range interactions, then that local time-varying field would look a lot like an external varying field.
Right.
But you're saying that your statistics can really distinguish between those two cases?
I didn't show you anything here, but yes, I think we can. And the idea here is, well, that's not yet finished work with David Schwab. We've been doing this like for a decade, just on and off, but at some point probably we'll finish it. So again, the example of an all-to-all Ising ferromagnet, right? What is the statistics of the mean field of the magnetization in the system? If you are away from the critical point, the magnetization is the average of magnetizations of individual units. Law of large numbers applies. Variance of magnetization is going to go down as 1 over n. At a critical point, variance goes, which is maybe not obvious, but it's a simple computation, variance goes as 1 over square root of n. So the standard deviation goes as 1 over quartic root of n. So notice that this variance is n dependent. Okay. And so if your collective variable is an emergent collective variable, then its variance or its probability distribution more generally should be system size dependent. If on the other hand, the emergent variable is externally driven, then it would be very weird if my brightness of the outside day depended on how many neurons I have in my brain, right? So I think that by actually inferring these collective modes using various machine learning tools, specifically we're using RBFs and so on and so forth, and then looking at the statistics of the emergent units, one can make a distinction whether these latent variables are externally driven versus emergent. But I think so. I don't know if that's the case. Right? Yes, Tim.
That was very nice. I think it's absolutely critical that we take the null hypothesis seriously, and so I'm super glad that you're looking at it so carefully. I still want to sort of throw out a, what if we're studying something that's slightly different than we think we are? Like, for example, what if this essentially emerges more as a consequence of our own ignorance than as a consequence of what's actually important and happening in, say, a neural circuit? And let me sort of give you an example of that. If you imagine, in fact, that circuits have very precise wiring, but that they are non-local, then you might imagine that the most important interacting partners for any given neuron are not anywhere in your recording, right? They're somewhere else in the brain that you're not observing in a sort of thorough, exhaustive sense. And so in a sense, I guess the counter to this very beautiful analysis, might it be that as you sort of renormalize away the scale of individual actors, that you are more and more left only with the sort of the fact that we're missing so much still, despite the huge data volumes we're getting, it's still dwarfed, compared to the scale of the brain as a whole, right? Is that something that keeps you up at night, or should, maybe it's not even something we should worry about, because it's still useful to know where our observations come from, and if the answer is our own ignorance, then that's something important to know.
Again, I think it's an absolutely correct point, right? And it does keep me up, maybe not at night, but when I get to work and I start thinking about this. But a totally different body of work that we have, which is, I didn't even start talking about it today, which is if you have this large dimensional time series, can you understand that this time series are missing something? Or to which extent they're missing something? And this all boils down to sort of older school Tachan style delayed embedding analysis and trying to but then putting on top of it modern neural networks. But yes, we're actively trying to understand how do we know that what we are observing, it does not come from ignorance rather than from real stuff. And I just didn't talk at all about it today.
Very cool.
Given the time, I think we should let people go and thank Ilya for a very interesting talk.